this is going to be for chapter 3. So for right now, I want you to know that we're skipping chapter 2. Chapter 2 is chemistry, water, properties of water. Those are things that you should already know coming into this class. So this is really just a placeholder to remind you that we're skipping that. You should already know that because of the prerequisites for this class. Okay, so chapter 3 covers microscopy. So one of the first things it talks about are different types of microscopy. So we used in micro, what we're going to use is we're using light microscopy. Okay, so light microscopy, but light microscopy was, or not light microscopy, electron microscopy was developed in 1932. So with it, instead of using light to observe the object, what we use are electrons. So you have to have a source of electrons. Here are 2 links to videos that describe how electron microscopy works. I'll let you guys click on those. There are 2 broad types of electron microscopy. So one is called TEM, or transmission electron microscopy. This is an image of it. So it magnifies things 10,000 to 500,000 times, but it gives a very flat and very detailed image. So this is the nucleus in here. This is supposed to be endoplasmic reticulum. So you can't see that with a light microscope. But you can see it with electron microscope, especially TEM. Okay, the second kind of EM that we're going to talk about is scanning. So scanning, you could say up to 50,000x. So what's really, really cool about it and makes it really neat is that it's 3-dimensional. So here's the head of a dissecting needle, and then as you get closer and closer and you increase magnification, you can see the bacilli. So you can see the little, it's actually E. coli on there. Okay, so nothing that we look at is actually colored. So the colors are all artificial and they're put in after the fact. So someone goes back in and edits the images. I have a blank slide. Keep going. All right, now fluorescence microscopy. So, I mean, typically we're using light microscopy, right? So the light's going to go through the specimen. So light is coming from the bottom and it's going up through. So in fluorescent microscopy, UV light comes down and it hits the specimen and it's going to excite the molecules that are fluoresce, and then they'll give off or emit light at a longer wavelength than what's exciting them. So the emission, this part, is what actually gets captured, and that's what's used to create an image. So there are some organisms that naturally fluoresce, so algae, plants will fluoresce. Some of them do. Some of them will fluoresce. And then there are other ways that we can modify things so that we can use fluorescence to see it. So, for example, this is a picture of cheek cells. So you guys probably remember scraping your cheek cell with a toothpick and putting it on a slide. Well, in this case, what we did is we scraped, these are somebody's cheek cells, and they stain them with a fluorescent stain. It's called DAPI. And the DAPI will actually bind to DNA and RNA. So it binds to the nucleus. So these bright circles in here, those are the nuclei inside the cheek cells, which is kind of cool. So we actually have this microscopes to do this at HCC. Oops. Okay, now one way that fluorescent microscopy is used is by doing antibody stains. So this is they're a diagnostic assay, and what we can do is we can make an antibody to an antigen that's found in an organism. So say you have E. coli. In theory, you could make an antibody that is specific to E. coli. So this is the antibody. Antibodies are Y-shaped proteins. We'll talk about that more. And they can be made to specific parts of things like bacterium. Okay, now what we could do is we can take this antibody and we can label it with the dye that will fluoresce. Okay, so we put the dye on the antibody. Then say you take a sample from someone, okay, so say you have, you're looking for E. coli in maybe meat or something, so you can make the slide, okay, and then you can add the antibody that's labeled, and then you can use fluorescent microscopy and you can see where the organism is that matches your antibody. So if they don't match, the antibody's not going to bind. Okay, so there's two different ways. The bullets get messed up with this when I import the PowerPoints into the program I use to make the videos. The bullets always look awful. I spend a whole bunch of time fixing them and then they always look the same. Okay, so how do we make slides for light microscopy? So in the lab, so far we've already done one kind of slide. So we've done a wet mount. So a wet mount or a wet prep, all we're going to do is we're going to take a drop of liquid and we place it on the slide and we put a cover slip on top of it. So it's really easy. The organisms are still alive. They're not dead, but you can't store it. And it could be a problem, right, if the organisms are still alive and they're bad, right? You don't want to be exposed to them. You don't want them living. So it's really, really easy. That's what we did with the pond water. Okay, now a dry mount is what we'll do this week in lab. So you place a little drop of water on the slide, and then we add a really thin smear of a small amount of your bacteria or whatever your organism is, and you smear it into the water. And then you let it air dry, and then after it's dry, we heat fix it. So when you heat fix it, what you're doing is you're passing it through heat. So the idea behind the heat fixing is that it's going to make the specimen stick to the slide. It kills the organisms, and it makes them so they'll take up the stain. They'll pick up the stain. So a disadvantage is it takes more time, and it does kill the organism. So if you want it to stay alive, that's not the way to do it. But we'll do this in lab. So don't worry too much about it. You'll understand how to do it when you learn how to do it. Okay, now we'll do lots of staining. Okay, so stain, sometimes it's referred to as dye, and dyes bind to cellular structures. So in microbiology, most of the stains that we use are cationic or are basic. So what that means is that they're going to bind to the cell components that are negatively charged. So the anionic cell parts. So some examples of stains that we'll use are methylene blue, which would look blue, crystal violet will look purple, and safranin, which will look red or pink. So a simple stain is a really easy stain to do. So it's one dye. It's very straightforward. You make a dry mount, or you do a smear, and then you just add the stain and rinse it off. So with that, you can figure out what shape the cells are and how they're organized or how they're arranged. In a differential stain, you use 2 or more stains. So you do kind of a series of stains and washes. And what they'll do is they're going to give you a lot more information, so it'll reveal a lot more about the structure of the cell, because different colors will bind at different times or to different structures. So the one that we're going to do all the time is called gram staining. This Zeal-Nielsen is actually an acid-fast stain. Whoops, that's supposed to say acid. An acid-fast stain. So we'll only do gram staining. That's the main kind of differential stain we'll do in class. You'll be experts at it by the time it's over. Okay, now, when we start looking at bacteria in the lab, there's certain terms that we're going to use to describe it. So in general, bacteria are between 1/2 and 2 micrometers, okay, or So it's a little u, it's a mu, big or small, right? They're very small. So what we're going to use oil to look at them, and we'll talk about the shape. Okay, so the shapes have different names, and we're going to use those names. The vocabulary is going to be really important. Okay, so cocci is plural, or coccus is singular, and that's a sphere. So there's a picture of it. Coxobacillus is kind of in between a sphere and bacillus, which is a rod. Okay, so it's in between. I'll help you with that one when we get to that one. Okay, Vibrio is shaped like a comma, so it's a bent rod. Spirillum is wavy, and then a spirochete is more like a spiral. And it's going to have this thing called an axial filament going through it, and that's a spirochete. So spirillum, I always think of it as more like waves, and spirochete is more like a spiral. Okay, bacillus is rod, cocci is spheres. So mainly we'll see cocci and we'll see bacilli. Okay, so bacillus is singular, bacilli is plural, coccus is singular, cocci is plural. Okay, now some organisms are what we call pleomorphic. So pleomorphic means that they're not all the same shape. So some of them are little, some of them will be like this, and some of them will be bigger. So they're different sizes. The only thing that can be that way are bacilli. We don't have pleomorphic spheres. Okay, now, the next thing we use to describe bacteria is how the cells are actually arranged. Like, how are they put together to create a colony? So, random is they're everywhere. They're just kind of, they're kind of randomly arranged, so there's no pattern there. That's random. Strepto means they're in strips. Okay, so here's strepto. I always think of them, students say strips because it makes it easy to say strepto. Chains is the way the book describes it. So strepto and strips, same thing, chains. Staphylo are clumps or clusters. Okay, cocci are the only thing that are staphylo. You'll never have a staphylobacillus. You only have staphylococci. Diplo, that one's pretty straightforward, right? There's two cells together. So here's diplo. Tetrad means you have 4 cells together. And then sarcinae means you have 8. So you have to know these. We will use those to describe cultures in lab. So yeah, you have to know the shapes, which is morphology, and the arrangements. You got to know those. Okay, now we're going to talk about Gram staining. So Gram staining is differential staining. So what it does is it's going to tell us about the cell wall of the bacteria. So there are two results that we could get. We could either get Gram positive or we could get Gram negative. So it's all about cell wall structure, which is in chapter 4. So Gram positive bacteria are always going to be purple after Gram staining. Gram negative are going to be red. And I know it's going to really look pink, but in order to not confuse you with the P, okay, we use the word red. So Gram positive and purple go together, positive purple, and then negative and red just makes it easier to remember. Okay, now the steps in making a Gram stain, and we'll do this in lab, is the first thing is you'll make a smear and you'll heat fix it. Then we'll add crystal violet, which is purple. Then we'll add a mordant. And a mordant is something that fixes a dye. Okay, or fixes a stain. So it makes it hang on to it. The mordant that we're going to use is grams of iodine. Okay, so if you think about like tie-dye t-shirts, if you tie-dye a t-shirt with RIT dye, the RIT dye doesn't have a mordant, and the t-shirts are treated with a mordant. So the color fades. It doesn't stay like a real nice, intense tie-dye. But if you buy like a professionally done shirt, they treat the cotton in the shirt with the mordant, and the mordant causes the dye to stay in there. So it fixes the dye. So that's what grams iodine does. Grams iodine, if you see it in the bottle, it looks kind of like this orangey brown. Okay, then we'll decolorize. So we're going to add alcohol, or it's 95% ethanol, to it. And then we'll do a secondary stain. So we'll go over this and I'll demonstrate it in lab. So you'll know how to do this by the time everything's done. Okay, so here's what it looks like. Okay, so if you have a gram-positive organism, you do the smear and then you fix it, and you add crystal violet. If you have a gram-negative, you add crystal violet, so everything looks purple. Then we add iodine. Okay, and then what we do is we're going to wash it with ethanol, so we're going to decolorize. The ones that are gram-positive, the purple is going to stay in the cells. The ones that are gram-negative, because of the way their cell wall is, the purple washes out. Okay, now because these look clear, right, they're really hard to see. You can't, there's no contrast. So what you have to do is we have to counterstain. And the counterstain is red. So these clear ones will take up the red stain and they'll look red, whereas the purple ones just stay purple. But we'll do it in lab, make sure everybody gets it. You'll do it a lot. You'll be so tired of me saying do a gram stain. Okay, now, oops, that's kind of messy. Other differential stains. So one of the stains that's used, it's called acid fast. I don't know why that looks so bad. Okay, so an acid fast stain. So acid fast is used to stain these organisms called Mycobacterium. So that's a genus. Mycobacterium can cause leprosy. It could cause TB or tuberculosis. So it's different than a gram. You can't gram stain it because this organism has a lot of lipids in the cell wall. So those cell walls really waxy. So the gram stains don't stick to it. So what they do is they do this acid fast stain. So they add a stain called carbofuchsin, which is fuchsia colored, and hydrochloric acid to wash the wax off. And so the bacteria will then end up being pink. So this is a sputum sample from somebody with TB. Okay, and you're looking for these purple cells, or pink cells in there. That's the organism. Okay, so they're not really water soluble, so the acid's going to help. Okay, spore stains. So we have to talk about endospores, but some bacteria produce these things called endospores or spores. And what they are is they're a resting structure. So when some bacteria get stressed out, in order to save themselves, what they do is they make these resting structures called spores. So they're different than spores in like mushrooms or fungi. They're resting structures. So they'll hang out. They can exist for like 10,000 years. They'll live for a really, really long time, or hang out for a really long time. And then if the environment becomes good again, they'll start to grow again. So this is in chapter 4 too. Okay, so spores, in our lab, we just stain them with the simple stain. So we just add like methylene blue to the slide. And the spores, because they have this thick outer wall, they just hang out. And they don't pick up a stain, so they look clear. Now with the spore stain, which is this differential stain, they actually will look green. So these little green things in here, those are the spores. Okay, a negative stain is when you're staining the background. Okay, so we're staining the background of it. So here's the cells. Let me change the color. These are the cells, right? And then here's the background. And then this bacteria has a capsule around it. And the capsule, we'll talk about in the next chapter, the capsule is just kind of like a coating of carbohydrates and proteins that are around it. And it doesn't pick up the stain. The capsule has to do with pathogenicity, and I'll talk about that more in the next chapter. But a negative is the idea is we're staining the background of it. Flagella stain is this stain that's specific just to flagella. We try it. It's really hard to do. It's easy to break off the flagella from the organisms, and it's hard to see it. But it's interesting. I guess if you had enough practice, you could get to do it. So that's what we need to cover for chapter 3.